Hello everybody, Mrs. Nest-Eden here. Welcome to the last lecture of this trimester. Today we're going to talk a little bit about cell structures and functions. Think about what you did yesterday. You started reviewing some of this. Today we are going to go a little bit deeper and talk about how all those organelles work together and talk a little bit about some structures that you may not know quite as well. First, just to review a little bit, there are some differences between animal cells and plant cells. All of the cells we're talking about today are eukaryotic cells that have a membrane enclosing the genetic material in a nucleus, but animal and plant cells have some different structures. Animal cells have some organelles called centrioles. They're in a region known as a centrosome. That's just a portion of the cytoplasm. And it's the part that helps to build the cytoskeleton. And they contain these organelles called centrioles that help organize the cytoskeleton, especially during cell division. Those are not present in plant cells. So that's one difference. Animal cells also sometimes have extensions called cilia or flagella or microvilli, depending on their size and their function. Those can be used to help the cells move or to stick to other surfaces. Plant cells, on the other hand, have a very large central vacuole. That's this big purple thing right here. Animal cells do also have vacuoles, but they don't have this really large one in the middle that provides some additional support and strength to plant cells. They also have chloroplasts, which perform photosynthesis, and some other organelles that fall into a group called plastids. Chloroplasts are just one type. Plant cells also have a cell wall outside of their cell membrane. All cells have a cell membrane. So in this picture here, the cell membrane is the yellow line, but plant cells also have that green cell wall outside, again, to provide some additional strength and support. Plant cells also have some special channels. We're going to come back to these a little bit later, but you can see these holes here called plasmodesmata. They're channels through the cell wall that actually connect plant cells to each other. So we'll come back to that towards the end, but those are a couple of the differences between animal and plant cells. One thing that you will notice as we go through the presentation today, anywhere that there is a yellow star, that actually means there's some kind of video or animation linked in that spot. I'm not going to show all of these to you right now because I don't want to make this Edpuzzle exceptionally long, but I'll link some of them in the notes of this Edpuzzle with a little bit about what they are, so you can decide which ones are most valuable for you to watch. The first set of organelles we're going to talk about are the endomembrane system. So if you think of a system that's like your digestive system or something, it's a bunch of pieces that all work together. Within a cell, organelles don't do their job on their own. And a lot of the major functions of cells involve this series of organelles that we refer to as the endomembrane system. Endo means inside, and membrane obviously means that they're made up of a lot of membranes. And you can see kind of a list of them up here. These organelles do a variety of tasks and work together to accomplish a variety of tasks. One of the major ones that you're probably most familiar with is the synthesis and transport of proteins. So you can see here, the purple in this picture is the nucleus that holds the instructions for making proteins, that DNA. It is attached, like literally the membrane is connected from the outside of the nucleus to the rough ER with all of these blue bumps. Those are the ribosomes, and the smooth ER back up here at the top. Those membranes are all interconnected. And then little vesicles bud off of the ER and bring things over here to this stack of membranes. That's the Golgi apparatus or the Golgi complex. And vesicles can bud off of that as well, that either become lysosomes or become vesicles that can transport things out to the cell membrane around the outside. And some of the substances that are transported through this system are proteins. That's one of the major things that cells do, is build proteins for a variety of functions. This system is also involved in metabolism. Remember, the word metabolism, we often think of it in terms of digesting food, but it really just means building up and breaking down molecules. So building proteins, building lipids are part of the metabolic reactions that occur within your cells. Lipids are made by the smooth endoplasmic reticulum, so they are also transported through the system. And all of these membranes, of course, are made of lipids. The smooth ER also detoxifies poison, so that's another function of this system within your cells. All of these membranes, as you can see, as we talked about, are related either through direct contact or by these vesicles. And so little bits of membrane, think of it kind of like bubbles that are kind of breaking off and then fusing back together. Those lipid layers can just butt off of each other and then rejoin and become part of each other. There are some differences in the chemical composition and the thickness of these different membranes and these different organelles, but they are all interconnected and work together to do a lot of the work that your cell does. A couple other organelles here are the lysosomes. You saw they can bud off of the Golgi apparatus. They're filled with what are called hydrolytic enzymes. Take a look at that word for a minute. What does hydrolytic mean? You've seen this before. Do you recognize the word hydrolysis in there? Hydrolysis. Remember, hydrolysis is when you put a water molecule back into a macromolecule and break it apart. Lysosomes break things down. They break up these big molecules. Sometimes they can fuse with what's called a food vacuole to digest or hydrolyze molecules for energy. So you can see in this picture in the upper right-hand corner, if your cell takes in some particles like this big gray food vacuole, lysosomes will come and the membranes will fuse together so that any digestive enzymes, those red dots within the lysosome, then are released into the food vacuole and hydrolyze. They break down those molecules and digest food that your cell can then use to create energy. This is also how white blood cells take in and kill bacteria or other pathogens in your body, and they also recycle damaged organelles and reuse those molecular components. Vacuoles are also related to these. Vacuoles are similar to vesicles, but they tend to be larger and they have some different functions. Some of them are those food vacuoles that can hold big particles that are brought in by your cells. Some of them are what are called contractile vacuoles, especially in kind of those single-celled creatures we saw in the pond water earlier in the year. And those help to pump water out to regulate the osmotic pressure in those unicellular eukaryotes. Some plants and fungi also have vacuoles that carry out enzymatic hydrolysis, similar to a lysosome. They can break things down. And plants will also use these to store things, small organic molecules. They'll kind of sequester toxins there to keep them in one spot. But then if anything eats the plant, they get those toxins in them. So it's a defense mechanism. They can also store pigments or color molecules in those vacuoles as well. The central vacuole in plants that we referred to before is much, much bigger. You can see in this picture, it takes up a huge amount of the area within a plant cell. And the point of that, the central vacuole is usually filled with water. And so if you imagine it like a water balloon full of water, it provides some extra support to that plant cell to help plants stay up even though they don't have a skeleton. The next set of organelles we'll talk about are things related to genetic instructions. The nuclear envelope is what you call the membrane that surrounds the nucleus. It's a membrane just like a cell membrane. It's a lipid bilayer. It's got little pores in it, holes that things can go in and out of, so those instructions can travel in and out of the nucleus. The DNA in the nucleus is organized into chromosomes, so it's kind of twisted up and wound around some proteins. But most of the time, it's not that organized. It organizes itself like that when the cell's going to divide. Most of the time, it's just kind of loose and spread out in a form we call chromatin. There's also another organelle within the nucleus called the nucleolus. And what it does is it builds ribosomes. Ribosomes are made of ribosomal RNA. That's that little R right here. rRNA is ribosomal RNA, and proteins build the components of ribosomes. So ribosomes are actually built in the nucleolus, and then they are sent out of the nucleus. Ribosomes aren't actually considered true organelles because they don't have a membrane around them. They're just made of ribosomal RNA and proteins. They're kind of categorized into two sections depending on where you find them. Free ribosomes are the ones that are just floating around out in the cytosol, And they make proteins that are going to stay within that cell, stay in the cytosol. They might act as enzymes to carry out some chemical reactions. They might become part of the cytoskeleton within that cell, but they're things that are actually going to be used within that cell. The ribosomes that are attached to the endoplasmic reticulum are called bound ribosomes. They're the ones that are making proteins either to become part of the membrane. So if you remember when we talked about cell membranes, all of those transport proteins and those proteins that are embedded within the cell membrane, those are made in the endoplasmic reticulum by those bound ribosomes. Or those bound ribosomes also make proteins that are going to be exported out of the cell. If it's, say, a cell that creates digestive enzymes that need to be released into your stomach or released into your saliva, those are also made by ribosomes on the endoplasmic reticulum. There's an article linked on the side here that gives you a chance to make some connections between some of the things that we've talked about. I'm not assigning this, but it's a really good idea to make sure that you understand how some of the things we're talking about now relate and connect back to the origin of life that we talked about before, and how they can give us clues to how living things are related. So take a couple minutes and read through and just think about some of the questions in that article. Now, briefly, we're going to touch on the organelles involved in energy. We're going to come back and spend like a whole unit with mitochondria and chloroplasts later in the year. Mitochondria are found in nearly all eukaryotic cells, and they obviously take glucose and break it down to make ATP energy. And you can see the inner membrane is very, very convoluted and folded. That increases the surface area because the ATP is actually made on that inner membrane. Chloroplasts are found in plants and some algae. They also have some compartments within them that allow for photosynthesis to occur. Chloroplasts, of course, absorb sunlight and use that energy to create glucose, which is then sent over to the mitochondria and broken down into ATP. There's also another organelle that's similar, it's kind of related to these two, called a peroxisome. This isn't one you need to know a lot about, but I put it in here just so you realize that even with all of 10th grade and this class, there's still organelles that you don't really know very much about. A peroxisome detoxifies hydrogen peroxide. If you remember a while ago, we did that lab with the enzyme catalase that breaks down hydrogen peroxide. Peroxisomes are where that reaction happens. So it detoxifies that byproduct of enzymatic reactions. Next, we're going to talk just briefly about structure and motility. The cytoskeleton is a structure that allows your cell and organelles to move. If you think of things like an amoeba that changes shape, or as your cell needs to kind of stretch and pull apart and divide into two cells during mitosis, the cytoskeleton is what allows it to change shape like that. It also helps organelles to move around. Vacuoles and vesicles don't just float randomly through your cell. They actually travel along the cytoskeleton and use that kind of like a highway through your cells. There are different structures that make up the cytoskeleton, and they have slightly different functions. Microtubules are one of those structures that grows from that centrosome region. That's where the centrioles are located. And the centriole's job is to help organize that part of the cytoskeleton during cell division. Some of those motor structures, like flagella and cilia, also contain cytoskeleton that helps the cells to move or to signal to other cells. Now we're going to move outside of the cell. And this is the part that may be new to you. These may be things that you haven't talked or you haven't learned about very much, are structures that exist outside of cells. First, we'll talk a little bit about plants. You know that plants have a cell wall outside of their cell membrane. Plants, prokaryotes, so that's bacteria, and fungi, and some unicellular eukaryotes all have cell walls. They're made of different things, though. Plant cell walls are made of that polysaccharide called cellulose that we've talked about before. Bacteria cell walls are made of something called peptidoglycan. That's another type of structural polysaccharide. And fungi, mushrooms, molds, mildews, things like that, make their cell walls out of something called chitin. So they all have cell walls, but they're not the same. They all differ in their chemical composition. There are also connections, like we mentioned before, between the cell walls of plants. In this picture, you can actually see the plasmodesmata, kind of these little tunnels from one plant cell to the next, right through the cell walls. These actually are lined with cell membrane, and they have a little bit of cytosol going through them. And so small molecules and water can diffuse directly from one cell into the next if they're small enough to go through those channels. Hang on. All right. Animal cells also have structures outside of the cell membrane. It's not a cell wall, but there are still structures that help keep our cells where they're supposed to be and help them to communicate and send signals to each other. And in animals, that's called the extracellular matrix. We've already talked a little bit about glycoproteins, these proteins that are bonded to carbohydrates. Glycoproteins are the primary molecules that make up the extracellular matrix. You can see it's kind of a combination of carbohydrates and proteins bonded together that make up this mess of fibers outside of the cell membrane. Collagen is the main structure within the extracellular matrix. That's these big, wide, rope-like fibers here. And there are others that kind of wrap around them. They're bonded to proteins within the cell membrane that are called integrins. And you can remember that because what those proteins do is they connect the cytoskeleton, they're literally attached to the cytoskeleton inside the cell, and the extracellular matrix outside the cell. And so they integrate those two structures, and signals can actually be sent from one to the other. So this is a way that cells can kind of talk to each other and regulate their behavior. If one cell kind of sends out a signal, it can be detected by other cells nearby through this network. There are also ways that animal cells are connected to each other. Like we talked about the plasmodesmata in plant cells, there are connections between animal cells as well. There are kind of three types that we're going to talk about today, and they all have slightly different functions. The first is what's called a tight junction. Think of this one, whenever I see this one, I think of this as literally stitching the membranes of these cells together. This forms a really tight seal between these cells. And so this is the type of junction you'll find anywhere that it's really important that nothing can sneak between the cells. So if you think about like your skin or the lining of your stomach, it's really important that things don't just seep out into the world between the cells of our skin. And so those cells are all connected with tight junctions. The second kind is called a desmosome, and this is actually a protein in the membranes that is also connected to filaments of the cytoskeleton. And you can see this desmosome in this picture stretches in and actually kind of knits those two cells together and reaches way into the cells. This makes really strong connections for things like muscle tissue that needs to be knit together really strongly. The third type are called gap junctions, and these are the ones that are most similar to those plasmodesmata in plant cells. They're actual little channels of cytoplasm that connect animal cells, and they do allow small ions or water molecules or really little things to diffuse from one cell to the next. So this is a way that cells can send signals to each other directly from one cell to the neighboring cells. So that's all for the new material for trimester 1. Couple of reminders are up there on the board. In the next 2 days, Wednesday and Thursday, I'm kind of giving you a little bit of flexibility so that you can decide when you want to take your last assessment. Tomorrow morning, there will be a final assessment posted in College Board. I say final assessment because it's the last one. It is not a final. It's the same size. It's about 15 points. Should take you around half an hour. It is, however, an assessment, so please sit down and take it all at once. Don't take it, start it now and then go back and finish it later. But you can decide whether you want to do that tomorrow or on Thursday, depending on what other tests, projects, and things you have to finish up. You can spend the rest of tomorrow and the next day finishing up the work that you have remaining in this trimester. Come in and ask me any questions you have. And just a note, I will try and remind you of this again. I know there's a material drop-off day on Thursday, and I would love to see your smiling faces, but there is nothing that you need to return to me for this class. You can keep your textbooks for AP Bio because obviously we get to stay together for the rest of the year. So I'll get your textbooks from you in May. If you have any questions, let me know. Have a great rest of your day.